everyone, my name is Anna Fernandez and my topic for presentation is Tandem Mass Spectrometry. What is tandem mass spectrometry? Tandem mass spectrometry is a technique to break down selected ions into fragments. The fragments then reveal aspects of the chemical structure of the precursor ion. In simple words, tandem mass spectrometry is a two-step technique used to analyze a sample either by using two or more mass spectrometers connected to each other or a single mass spectrometer by several analyzers arranged one after the another. Principle of tandem mass spectrometry. The molecules of a given sample are ionized and the first spectrometer MS1 separates these ions by their mass to charge ratio M by Z. Ions of a particular M by Z ratio coming from MS1 are selected and then made to split into smaller fragment ions by collision induced dissociation. The fragments are then introduced into the second mass spectrometer MS2, which in turn separates the fragments by their M by Z ratio and detects them. The fragmentation step makes it possible to identify and separate ions that have very similar M by Z ratio in regular mass spectrometer. Structure. Tandem mass spectrometry includes triple quadruple mass spectrometer, quadruple time of flight, and hybrid mass spectrometer. Tandem triple quadruple mass spectrometer. Triple quadruple mass spectrometers use the first and third quadruples as mass filters. When analyzed past the second quadruple, the fragmentation proceeds through collision with gas. Usually, triple quadruple mass spectrometers are used for pharmaceutical industry. Quadruple time of flight. Quadruple time of flight mass spectrometer combines TOF and quadruple instruments which cause high mass accuracy for product ions, accurate quantitation capability, and fragmentation experiment applicability. Hybrid mass spectrometer. Hybrid mass spectrometer consists of more than two mass analyzers. Tandem MS instrumentation. Multiple stages of mass analysis separation can be accomplished with individual mass spectrometer, elements separated in space or using single mass spectrometer with the MS step separated in time. Tandem in space. In tandem mass spectrometry in space, the separation elements are physically separated and distinct. Although there is a physical connection between the elements to maintain high vacuum, these elements can be sectors, transmission quadrupole, or time of flight. When using multiple quadruples, they can act as both mass analyzers and collision chamber. As you can see in figure, there are three quadrupoles, quad 1, quad 2 and quad 3. They are aligned in a row. Precursor ions are selected in quad 1 and sent to quad 2 for dissociation, and the general product ions are sent to quad 3 for mass scanning. Quadruple time of flight. By doing tandem mass spectrometry in time, the separation is accomplished with ions trapped in the same place with multiple separation steps taking place over time. A quadruple ion trap of FTICR instruments can be used for such an analysis. Trapping instruments can perform multiple step of analysis, which is sometimes referred to as MS to the N. As shown in diagram, the quadruple time of flight mass spectrometers uses a quadruple, a collision cell, and a time of flight unit to produce spectra. The lighter ions accelerate faster down the flight tube to the detector, thus determining the ion mass to charge ratio. Quadruple time of flight mass spectrometers Systems have MS-MS capabilities and can be coupled to GC or LC system. Hybrid ion trap or FTMS. For the hybrid ion trap or FTMS, instruments precursor ions are selected and fragmented in an external ion trap. The generated product ions can be detected either in the external trap or by FTMS. Fragmentation. In MS, fragmentation is the dissociation of energetically unstable molecular ions formed from passing the molecule in the ionization chamber of a mass spectrometer. Fragmentation of gas phase ions is essential to TMS and occurs between different stages of mass analysis. There are many methods used to fragment the ions, and this can result in different types of fragmentation and thus different information about the structure and composition of the molecules. In-source fragmentation. Often the ionization process is sufficiently violent to leave the resulting ions with sufficient internal energy to fragment within the mass spectrometer. If the product ions persist in their non-equilibrium state for a moderate amount of time before auto dissociation, this process is called metastable fragmentation. Nozzle schema fragmentation. Nozzle schema fragmentation refers to the purposeful induction of in-source fragmentation by increasing the nozzle schema potential on usually electrospray-based instruments. Collision-induced dissociation. Post-source fragmentation is most often what is being used in a tandem mass spectrometry experiment. Energy can also be added to the ions, which are usually already vibrationally excited, through post-source collisions with neutral atoms or molecules. The absorption of radiation or the transfer or capture of an electron by a multiple charged ion. Collision-induced dissociation, also called collisionally activated dissociation, involves the collision of an ion with a neutral atom or molecule in the gas phase and subsequent dissociation of the ion. Electron capture and transfer methods. The energy released when an electron is transferred to or captured by a multiple charged ion can induce fragmentation. If an electron is added to a multiple charged positive ion, the Coulomb energy is liberated. Adding a free electron is called electron capture dissociation (ECD), and adding an electron through an ion-ion reaction is called electron transfer dissociation (ETD). Similar to ECD, ETD induces fragmentation of cations by transferring electrons to them. Application. Peptides. Tandem mass spectrometry can produce a peptide sequence tag that can be used to identify a peptide in a protein database. When intact proteins are introduced to a mass analyzer is called top-down proteomics. And when proteins are digested into smaller peptides and subsequently introduced into the MS are called bottom-up proteomics. A notation has been developed for indicating peptide fragments that arise from TM spectrum. Fragments containing the N-terminus are labeled A, B or C depending on the site of the cleavage, whereas fragments containing the C-terminus are labeled X, Y or Z. The numbers indicate the number of amino acid residues in the fragment ion. Oligosaccharides. Oligosaccharides may be sequenced using tandem mass spectrometry in a similar manner to peptide sequencing. For oligosaccharides, fragments containing the reducing end are labeled X, Y or Z depending on the site of the cleavage whereas fragments containing the other end are labeled A, B, or C. The numbers indicate the site of the sugar residue. Y, Z, B, and C ions are fragments due to the glycosidic cleavages, whereas A and X ions result from cross-linked cleavage. Conclusion: mass spectrometry is a versatile technique that has found wide application in a wide range of fields. Modern tandem MS systems are robust and sensitive. Tandem mass spectrometry is highly informative for the mass determination of fragmented ions originating from peptides. Thank you.